Hello everyone, my name is Ashwini Singh and welcome to the part 2 of the Biochemistry Lecture 1 series, Foundations of Biochemistry. In the previous video, we learned about how the creation of universe led to the formation of chemical elements and eventually life. We also looked at different features of living organisms and how biochemistry describes the structures, mechanisms, and chemical processes shared by all organisms. Today, we'll learn about the backgrounds of biochemistry. So, biochemistry is based on five foundations: cellular, chemical, physical, genetic, and evolutionary. Let's begin with biochemistry at a cellular level. So we all know there are two types of cellular organisms, unicellular and multicellular. So unicellular organism consists of single cells and are microscopic, while the multicellular organisms are larger and have different types of cells which vary in size, shape, and have a specialized function. However, all the cells share some fundamental properties which can be seen at the biochemical level. So let's dive deeper into this. So we all know that cells are the structural and functional unit of all living organisms. So the plasma membrane defines the periphery of the cell. The plasma membrane is composed of proteins and lipid molecules that form a hydrophobic barrier around the cell. This barrier gives free passage to the inorganic ions. There are two types of proteins in the plasma membrane, the transport proteins and the receptor proteins. These transport proteins help the inorganic ions to pass through the hydrophobic barrier, while the receptor proteins transmit signals into the cells. Since the individual lipids and proteins of the plasma membrane are not covalently linked, the entire structure of the cell is remarkably flexible, allowing changes in the shape and size of the cell. Plasma membrane encloses the internal volume of the cell, which is the cytoplasm. It is composed of an aqueous solution, the cytosol, and has a variety of suspended particles with specific functions. These particles are membranous organelles such as mitochondria and chloroplasts in cells or endoplasmic reticulum, or it can be supramolecular structures such as ribosomes and proteasomes. The cells also have either a nucleoid or a nucleus. Nucleoid is present in bacteria and archaea and is not separated from the cytoplasm by a membrane, while nucleus is present in eukaryotes and is enclosed within a double membrane nuclear envelope. Bacteria, archaea, and eukarya are three domains of life to which all organisms belong. The single-celled microorganisms are distinguished on genetic and biochemical grounds into bacteria and archaea. Bacteria inhabit soils, surface waters, and tissues of other living or decaying organisms, while archaea inhabit the extreme environments like salt lakes, hot springs, highly acidic bogs, and ocean depths. All eukaryotic organisms make up the third domain, which is eukarya. So, all organisms require a source of energy to perform cellular work and differ widely in its sources and biosynthetic precursors. There are two broad categories of organisms based on energy sources. One is phototrophs and the other one is chemotrophs. So, phototrophs trap and use sunlight as their energy source, while chemotrophs derive their energy from oxidation of a chemical fuel. Phototrophs and chemotrophs may be further divided into two categories: autotrophs and heterotrophs. Autotrophs synthesize their biomolecules directly from carbon dioxide, while heterotrophs require organic compounds made by other organisms. Phototrophs use water to reduce carbon dioxide. It performs oxygenic photosynthesis. If it does not, it performs anoxygenic photosynthesis. Some examples of photoheterotrophs are green non-sulfur bacteria, purple non-sulfur bacteria. Some examples of chemoautotrophs are hydrogen, sulfur, iron, nitrogen, and carbon monoxide oxidizing bacteria. If the final electron acceptor is oxygen, the examples are animals, fungi, protists, and bacteria. If oxygen is not the final electron acceptor, then for organic compounds, the example are fermentative bacteria such as lactobacillus. If inorganic compounds are used, then example is Pseudomonas tenacious. Bacterial and archaeal cells share common features but differ in important ways. The best studied bacterium, Escherichia coli or E. coli, has a protective outer membrane and an inner plasma membrane that encloses the cytoplasm and the nucleoid. Between the inner and the outer membranes is a thin wall of peptidoglycan that gives the cell its shape and rigidity. The plasma membrane of the bacteria consists of a thin bilayer of lipid molecules penetrated by proteins. Archaeal plasma membranes have a similar architecture, but the lipids can be strikingly different from those of bacteria. Some bacteria are called gram-positive because they are colored by gram stain and have a thick layer of peptidoglycan outside their plasma membrane, but they lack an outer membrane. Gram-negative bacteria have an outer membrane composed of a lipid bilayer into which are inserted complex of lipopolysaccharides and proteins called porins that provide transmembrane channels for the diffusion of low molecular weight compounds and ions across the outer membrane. The cytoplasm is organized by cytoskeleton and is highly dynamic. Fluorescence microscopy reveals that several types of protein filaments crisscrossing the eukaryotic cell forms an interlocking three-dimensional meshwork, the cytoskeleton. Eukaryotes have three general types of cytoplasmic filaments: actin filament, microtubules, and intermediate filament. These have different width, composition, and have specific function. All these types of filaments provide structure and organization to the cytoplasm and shape to the cell. The actin filament and the microtubules help to produce the motion of organelles or of the whole cell. Each type of cytoskeletal component consists of simple protein subunits that associate non-covalently to form filaments of uniform thickness. These undergo constant disassembly into their protein units and reassembly into the filaments. The assembly, disassembly, and location of all types of filaments are regulated by other proteins, which serve to link or bundle the filaments or to move cytoplasmic organelles along the filaments. The cytoskeletal proteins also serve as rails along which the cellular organelles move throughout the cell. Their motion is powered by energy-dependent motor proteins. This structural organization of cytoplasm is far from random. The motion and positioning of organelles and cytoskeletal elements are under tight regulation, and at certain stages in its life, a eukaryotic cell undergoes dramatic, finely orchestrated reorganization, such as the events of mitosis. Interactions between cytoskeleton and organelles are non-covalent, reversible, and subject to regulation in response to various intracellular and extracellular signals. Cells build supramolecular structures. Macromolecules and their monomeric subunits differ greatly in size. For example, alanine, which is an amino acid, is less than 0.5 nanometer long. However, a molecule of hemoglobin, which consists of nearly 600 amino acid subunits, folds into a globular structure of 5.5 nanometer in diameter. The monomeric subunits of proteins, nucleic acids, and polysaccharides are joined by covalent bonds. However, macromolecules are held together by non-covalent interactions, which are much weaker than the covalent bonds. Among these non-covalent interactions are hydrogen bond between polar groups, ionic interactions between charged groups, hydrophobic interactions by nonpolar groups in aqueous solution, and van der Waals interaction. The large number of weak interactions between macromolecules in supramolecular complexes stabilize these assemblies, producing their unique structures. Next up, we'll be learning about the chemical foundations of biochemistry. That's all for today. We'll continue this in the next lecture video. If you like my content, please subscribe to this channel and get notified of the new videos.